looking at oncology or cancer as a disease. Maybe even 20, 25 years ago, a lot of times when you're diagnosed with this horrible disease, there are very few options. So these biologics really have changed the complete landscape of oncology. Really is not only just efficacy, but also the patient experience being on those therapies are very different. I would say these days compared to 20 years ago, the landscape is completely different. Oncology becoming a disease that can be managed rather than a death sentence. It is a combination drug, large and small molecule, but analytically is very complicated because it has both large and small molecule component.
Jim, what are your thoughts about this concept of gluing proteins together within tumor cells to induce cell death, and how might this strategy evolve in the future?
So very interesting you asked this question. Actually,
Welcome to the BioTalk Unzipped podcast, where we unzip the stories behind medical progress by sharing the latest and greatest advances in biopharmaceuticals and medical technologies in a fun, entertaining, and enlightening format. And now your co-hosts, Gregory Austin and Dr. Chad Briscoe. Hello and welcome to BioTalk Unzipped, where our mission is to unzip, unlock, and uncover the stories behind medical progress. I'm your host, Gregory Austin from Kansas City. I'm a life science professional in the quality and regulatory consulting space for ECI. Along with me from North Carolina is my friend and co-host, Dr. Chad Briscoe. Chad is a bioanalytical scientific expert with Celerion, and we publish BioTalk Unzipped twice a month and bring experts from the different corners of the pharmaceutical, biotech, and medical device worlds to share their innovations, insights, career journeys, and personal stories to the show. Please be sure to subscribe, like, and comment on our episodes, and let us know what you'd like to hear about in the future, and thank you for listening. Today, from Princeton, New Jersey, we are joined by Dr. Jim Shen, a highly respected thought leader in the bioanalytical field with over 25 years of experience. He currently serves as the Executive Director and Head of Regular Bioanalysis Operations at BMS, where he oversees complex bioanalytical operations, including method development, assay troubleshooting, and regulatory submission. Dr. Shen's experience spans mass spectrometry, automation platforms, and high-resolution HPLCs, contributing significantly to drug development. He's also the vice chair of the bioanalytical community at AAPS. A prolific writer with 45 publications, Jim is a key figure in advancing pharmaceutical research and innovation at BMS. Jim, welcome to the show. How are you?
Very good. Thank you very much, Greg. It's good to be on the show, as well as you. Hello to you, Chad.
Yeah, hello, Jim. Good to see you. Thanks for joining.
Good to see you.
So it's our tradition here at BioTalk Unzipped to make a $100 donation in our guest's name to the charity of their choice, and you have decided to give that to the American Cancer Society, which many of us can certainly appreciate. Why the American Cancer Society?
A couple different things, really, Greg. I think for one thing, my chosen field or my profession, the Bristol Myers Squibb and Company, really is very strong in the cancer field. So really, our pharmaceuticals has impacted so many patients' lives, and my job, a major part of it is actually in cancer. So seeing how the patients suffer from this horrible disease, really, I think we need all the help we can get. The other one, actually, I thought about this, the Alzheimer's Association. My father actually suffered from that. But, I think between those two, I have to pick one. So I went with the American Cancer Society.
Yeah, certainly understandable. Speaking of cancer, I'm going to come out hot of the gate with a BMS drug question. And this is about the immunotherapy combination drug Opdualag, if I'm saying that correctly, which is designed to enhance the immune system's ability to fight metastatic or unresectable melanoma. And just like you mentioned, we all have a story about someone we've lost or someone we're losing to some type of chronic or fatal disease. And my dad, I saw this online because it was recently accepted by the Scottish Medicine Association, and I did a little bit of research on it. He died 14 years ago from melanoma and started out on his back, and 7 years later metastasized in his brain. It took him pretty fast. And so it's near and dear to my heart, this type of a breakthrough. I'm curious, did you get a chance to work on this drug, and how do you feel about this as maybe a building block to extend life? Hopefully even beyond 21 months.
Absolutely, Greg. So Opdualag really is I would say, a fantastic medicine that really changes so many patients' lives. Interestingly, just looking at oncology or cancer as a disease, maybe even 20, 25 years ago, a lot of times when you're diagnosed with this horrible disease, there are very few options. You pretty much go on management therapy. Maybe there is a chemical therapy that goes in against the cancer, and it's also very, very aggressive. At the same time, you're suffering. So these biologics really have changed the complete landscape of oncology. Not only some of them have fantastic efficacy, really it's the not only just efficacy, but also the patient experience being on those therapies are very different. Biologics, you have to get infusion maybe once or twice a month or maybe more frequent, but really the side effect compared to just the chemical therapy are so much better. People can tolerate it so much better, so people can live really a more normal life. Really, if you look at some of the oncology, I would say drugs these days, so many new things are coming to the market, like ADCs, like a combination therapies, and all these compounds. Really, it's extending, I would say, patients' life months at a time. I'm reminded of a story. I think my wife has an uncle last year passed away through a CRC, a horrible disease.
Hmm.
And at the time, there were essentially no drug to treat that particular type of cancer. And also the drug did such a thing on his body. He at the end just gave up. He basically told me and my wife, we just don't want to do it because there's so much suffering seeing other people looking at me suffer. I just don't want to do it anymore. Then a couple months after that, my company actually came out with a new version of the Opdualag that actually had some indication toward that particular type of biomarker. So I just felt like even if people just had a couple months, maybe a new oncology drug will come on the market that would extend their life. So really, I would say these days compared to 20 years ago, the landscape is completely different. Oncology becoming a disease that can be managed rather than a death sentence.
And it's a really good point. And it kind of hit me right here when I read that, because I was like, oh, if it could just been a couple decades sooner, then
Yeah,
he
yeah.
would have bought maybe a little more time. So
I've certainly thought about that with loved ones that I've lost or have gone through cancer treatment. Is, man, if it would have been today, how different would that picture be? Or if it's 10 or 20 years from now, how different is that picture? And I think that's, Jim, what we're all working towards. And Greg, in your contribution to in your field, that's what we're working towards, right? We're working towards a better tomorrow. I think that's even one of the pharma companies. I think that's their line is something about a better tomorrow or something like that. That's what we have to think about all the time is we're doing the best we can every day to see a better tomorrow. I had a question since we're talking about this and with BMS. I remember, I don't know what it was, 15 years ago, something 20 years ago, BMS was only a small molecule company, and they added biologics, and they became a company that's now, I don't know what, 50-50, or certainly has a large portfolio of both biologics and small molecules. Can you talk about like and I know you were with a different company back then, I believe, but that transformation just in the industry from just small molecules to the biologics and the complex modalities today.
Absolutely, Chad. I think that's actually a very interesting transformation for BMS as well as the whole field. So when I joined BMS about 13 years ago, at that time is exactly the time the nivolumab or Opdivo is becoming
Hmm.
the drug that's going into submission. So at that time, a lot of research is being put into large molecule, and really those drugs, I would say, became a central, like very central to company success, the large molecule.
Hmm.
But
Yeah.
through these years with BMS and its various acquisitions, various transformations, for example, merging with Celgene, actually brought in a lot of new modalities. These day and age, actually, I would say we are pretty evenly split between small large molecule, and also small large molecules is really no longer fitting its traditional definition. Before, small molecule is anything, say molecular weight less than 500. But these day and age, really, you can have a combination of a large and small molecule that works together essentially as delivery mechanism. Then after you get to the cancer, the small molecule become a warhead killing the cancer cells, for example, like ADC, antibody conjugate. So
Hmm.
those becoming such a hot commodity across the entire industry. Every company is working on that. It's very difficult. I mean, the line, I think, is being smushed between a small and large molecule. So, Chad, you're absolutely right. Traditionally, 20, 25 years ago, everything was essentially small molecule. Then everything was a pure large molecule for a little bit. But this day and age, everything's about hybrid.
So another, just let me ask you to explain a little bit more about antibody drug conjugates because that's, we have a diverse, listeners that not everybody knows them, and you hit on it quickly, but to me, that's one of the most exciting and interesting types of molecules. And can you just explain that a little bit more for the listeners who don't know what an ADC is in
Absolutely.
layman's terms?
Sorry for interrupting you there, Chad, but
No, no, go
ADC
ahead.
really anti-drug conjugate is a class of biopharmaceutics designed to really target and treat a particular disease. Very often, this is targeting cancer, and unlike a chemotherapy, ADC really is a type of molecule that has a large molecule core, and schematically it is a large molecule, but conjugate associated to it what we call a cytotoxic payload or drug, and the drug will be given as if it's a large molecule drug and infused, but this does not have systematic effect on the whole body. It would act like a warhead and specifically going toward a particular part of the body where the antibody would hire the hire the drug onto,
Right.
then it would release its warhead or antigen to kill that particular cancer cell. So it's a designer drug. It is a combination drug, large and small molecule, but analytically is very complicated because it has both large and small molecule component. And hopefully, when we get to the podcast, Greg can find some type of schematic to kind of show
Hmm.
different part of ADC, including the antibody, the linker, and the cytotoxic drug.
Yeah, that's awesome. Thanks. Yeah, it's almost like a Trojan horse. Yeah, it's brings the fight right to the site of where the action's happening. That's awesome. Thanks, Jim.
That's excellent technology. I appreciate that explanation. So we were talking a little bit about oncology before. That brings us into our first new news section. New news.
New news.
New news. In an article published in August 12th by Jonathan Gardner in Biopharma Dive, Halda Therapeutics recently raised $126 million in new funding to advance its innovative cancer therapy platform called RipTac, which uses a dual-acting drug technology. So basically what RipTac does, it works by gluing a cancer-specific protein to another essential protein within tumor cells, triggering a protein interaction that halts the kind of vital cellular functions that leads to tumor cell death. This funding will help them bring their lead drug candidate into clinical trials by next year, targeting metastatic prostate cancer patients who have developed, resistance to hormone therapies. Halda's approach builds on early protein degradation research, but focuses on creating more selective and potent treatments. Jim, what are your thoughts about this concept of gluing proteins together within tumor cells to induce cell death, and how might this strategy evolve in the future?
So very interesting you asked this question. Actually, I'm actually not familiar with this company, but
Yeah.
actually the the concept this called the ligand directed degrading, and this is actually a big therapy area for BMS as well. So the ligand
Ah.
directed degrader really is what we call molecular glue. It's induces. I don't know if their technology exactly does this, but in our technology, essentially, is a bifunctional ligands. One side of it will target essentially cereblon binding moiety, and to induce proximity binding between the drug itself as well as the cereblon E3 ligase complex. The other side would bind a drug then what we generally call this, there are two different things. They are ligand direct degrader, and they are another type of degrader called, most of these are called PROTAC. And these type of molecules actually is really hot right now in the industry. Not only this is becoming, I would say, a designer type of degradation, not only for oncology, but for other type of disease area. Also, this is very interesting from a chemistry perspective, because the very beginning of, I remember this type of drug is like in the 1980s, the image was essentially discovered by a Japanese scientist. And the image actually has its, I would say, basis in the 1950s when a drug was synthesized for morning sickness in Germany. And that has two diastereomers. Then one of the diastereomers actually caused horrible deformities in babies. I think many people remember.
Thalidomide.
Yeah, so thalidomide. I mean, I think I am too young to remember that, but
Yeah.
I haven't seen the picture. But the thalidomide, very interestingly, so has basically two stereoisomers. Stereoisomer is basically a compound that mirrors each other. Very often, you cannot synthesize one without having the other.
Great left hand, right?
Yeah. So in this
Yeah.
case, one was curing morning sickness. The other one was essentially making baby deformities.
Yeah.
So these type of modality we actually support quite a bit in the lab. It's very challenging analytically to support it, because you have to characterize both of these isomers independently, and we have to separate them onto an instrument called HPLC, and we have to quantitate the different concentrations. And like Chad just said, like one could be toxic, so we had to figure out if one is now converting to another and all that. So it's very, very interesting to hear all these different modalities are getting funding from Wall Street, because there are just so many new drugs that's coming onto the scene. That's very different how the traditional drug kind of work, just the inhibition of a particular ligand or a pathway. These days, you have a degradation pathway, you have ADC, you have oligonucleotides, essentially disrupting the RNA actions. So a lot of these are really next generation pharmaceutics that pharmaceutical companies are working very hard with. And we really think this is going to really change how disease will be dealt with. And this is really going to have a positive impact on human health.
How far along the pipeline is the BMS version of this protein technology?
We actually are on something like third or fourth generation. So the initial compound actually is widely used in blood cancer, and that's already a drug. And the second generation is coming online, pretty much getting into patients' hand. And we in research area are working on third or fourth generations. Like I said at the very beginning, this is usually mostly used in oncology area, but I'm seeing other companies as well as other, I would say, even research institutions diving into other type of disease area above and beyond oncology.
Excellent.
That's awesome.
That's exciting to hear. Yeah. So, Jim, what inspired your journey to get into bioanalysis, and what keeps you motivated to grow in the field?
A couple of things, right? So actually, bioanalysis is kind of like, I would say, a byproduct just happened. Actually, my journey into mass spec a little bit more interesting. So,
tell me.
So my undergrad is with University of Arizona, and this is one of the interesting stories that you always do not want to prove your father right, but sometimes, unfortunately, that have tendency to happen. Now I'm a father of two grown boys. I start to be more appreciative of what a father goes through for their kids' professional career. So I always liked the computers. Even when I was in high school, I played with computers. I programmed. I was a nerd playing computers before it was fashionable. So I basically want to become a computer scientist. I said, I want to be a programmer, and I wanted to do this. My father, very traditional Chinese father, kind of like, computer, what is that? You don't want to be in that. So he was actually a professor at the University of Arizona at the time, and I basically said, I want to go into computers. And he said, you are getting essentially a lot of help here to go to college. So if you want to get help, you are also going to get a PhD in chemistry. I said, I don't want a PhD in chemistry. I want a PhD in computer. Then he basically said, oh, getting a PhD in computer is great as long as you also get a PhD in chemistry. So instead of paying my own way and having somebody pay my way to go to college, I said, sure, I will get a degree in chemistry. And after I started doing computer science for a couple years, so I essentially had to do major all the way until I was a senior. I actually carried on both majors. I was like, wait a minute, this is really not the life I want. I enjoy playing with computers. I actually don't enjoy programming because you're
Yeah.
staying up night after night, staring at the code and tearing heart out. I was like, this is not exactly what I envisioned being a computer scientist is about.
Yeah.
So
Yeah.
I ended up getting a degree in chemistry. I actually didn't finish or complete my computer science degree. So in that sense, I think, unfortunately, I proved my father correct. And I think a computer, just as a general knowledge and approach perspective, continuously has benefited me throughout my professional career. So I'm very grateful having that background. But this is one of the stories why I got it in chemistry. Then once I got into chemistry, I kind of thought because my father's organic chemist, so is my mother, I kind of like, I don't want to do this. I still need to rebel in some limited way. So I said, with my computer background, I want to go into analytics. So I went to analytical chemistry. I got into graduate school, University of Texas, Austin. I was like, this mass spectrometry, this is cool. A lot of computers, a lot of tweaking, a lot of working with big machines. I was like, I enjoy this. I want to do this. So at that time, my professor there, Dr. Brockbell, she's still there. She kind of like interviewed me and she said, Jim, you are aware that if you go into mass spectrometry, the only people that compare to you would be like the NMR scientists because there is no job other than being inside of either in like a national lab or be a professor because industries can't really afford mass spec. They don't really use it. Then, well, I was like, I'm fine with that. I just want to play with computers and play with the instrument. So I got into mass spec. About the third year in graduate school, this interesting thing called Jiangfeng and this interesting thing called electrospray happened. Then it gave us ability to look at biomolecules inside mass spectrometer. And now everything blew up. And I have been riding that wave continuously, and I have been a mass spectrometrist. So becoming a bioanalytical chemist, really, that's, I would say, almost an accident. I had a background in mass spec. So at the time, my wife had a good job in Austin, so I wanted to kind of stay in the area. Austin, Texas is not known for big pharma. So there's very few jobs. I interviewed, I remember Bayer and Stanley Neese, both in QC work and in chemicals. One of the job was actually climbing up to the conning tower, take a sample every hour and come back around in the lab. I was like,
That doesn't sound like you, Jim.
I was like, that doesn't sound like Jim, huh? So I was like, I need to look for something else. So one of my friends actually introduced me into this place, this CRO, and they said, what's a bioanalytical CRO? I was like, what's a bioanalytical CRO? There, he was like, don't worry about it. Even if I explain it, it may not mean anything to you, but they're looking for somebody who does mass spec.
Yeah.
So that's how I kind of got started. So kind of by accident. But really, in this field, I think Chad also probably Chad is one of the forefathers in bioanalytical chemistry, and he has been here for a long time. Really, after you get into the field, it's a wonderful field to be in. I was looking at the I think the Unzipped which grant you did.
With Russ Grant,
right? So he was talking about, and also really it's about a community and the people in it. I really feel the same way because in bioanalytical, although we are pretty big field now, everybody still knows everybody. That's really nice, and really the people in it is tremendous. And also, I'll say certain type personalities, a little nerdy, chemist type, biologist type, we will get well together. We will get along together very well. So
yeah,
it's good. Yeah, Russ had a funny story about how his love for mass spec was very similar, and he would study it, he'd practice it, he would break it, he would fix it and start over, and that's where he got started. That's a good story.
Yeah, and I think Jim, you talked about the early electrospray days, and there was a lot of challenges with those early instruments. Just getting, nowadays you just flick a switch and it's on, and you just tell it to tune itself, and it tunes itself. But yeah, back in those days, I had an instrument that it was a Vestec, so it was Marvin Vestal's instrument, and you had to it didn't have any like safety mechanism on it, so you would actually pull the 5,000 volt probe out, and you had to get the HPLC just a little tiny drop on it, and then you'd stick it in and turn the gas on at the same time, and then it would start spraying, and then you wanted to keep that thing going forever because you didn't want to have to pull that 5,000 volt probe out again. And so that was kind of my first electrospray mass spec experience at University of Michigan was using that old Vestec. So that was something.
Yeah, absolutely, Chad. And it's actually very ego bruising these day and age to be going to the lab and looking at the mass spec. It's probably akin to a new mechanic looking at cars. When you open it up, you don't really recognize
Yep.
anything.
Yep.
I was
Yeah.
just telling
Yeah.
the people in the lab, we used to we would put a rail, we would clean, we would essentially repair the
Yeah.
mass spec. We do all these things ourselves. So we really
Hmm.
had a good understanding of it. But these day and age, kind of like this black box. People just push a button and they expect it to work.
Yep.
And then when they don't
Yep.
work, they call their service engineers.
Yep.
So
Yep.
I guess that's maybe an evolution of old type
Yeah.
of modern
Yeah.
instrumentation. At the same time, for old school people like me, it's a little sad because
Yeah.
there is
Yeah.
I would say a degree of proficiency and a degree of knowledge that's lost. And
A depth of understanding that you get by really getting in and digging into the instrument. Similar. I think the car analogy is great, right? If you can get into a 1980s or earlier car, you're really going to know how that engine works. You're going to know what happens when you push the pedal, and then nowadays nobody even knows how to drive a stick. And
I
that's
I do not. So
yeah, it's fantastic.
So Jim, you mentioned technology, and I'm curious, how has the integration, or have you been involved with the integration of AI and mechanistic modeling that's been influencing the drug development processes at BMS?
absolutely, I think actually AI and machine learning right now is the buzzword across any industry. It doesn't matter which one it is. I think I get free books from Amazon Prime. If you look at the amount of free book that's recommended to you, that's AI machine learning. I would say probably populates 50% of the book that's being recommended. So, but it's kind of interesting because actually you start like we obviously want to ride this wave. We want to understand AI machine learning. We want to also be part of it. But really, there's a lot of buzzwords out there, not necessarily a lot of substance right now. So you can talk to a lot of people. You go, "Oh, this would be great if you can do this." Then people say, "Well, that would be great, but we don't really know how to do that." So we have approached our instrument vendors. We have approached our software vendors, and everybody knows how to use ChatGPT, which is great. And there is technology that, for example, people like uh, BMS work with these AI companies um, to get a version of those ChatGPT into our organization. It's a fantastic if you want to write an email or doing somebody's performance review. But if you want to get to the next level of AI, it really requires again a more in-depth understanding of computer science and also a expertise in the area you're working with. So that linkage hasn't really occurred because the really adept computer scientists said, "Oh, this is great. We gave you a ChatGPT model. Now you can do all these great things." But
Hmm.
if you want to go beyond just tap in, tell me a joke, then the application in analytical chemistry or in bioanalytical are still pretty far in between. We're working on certain things that's pretty revolutionary, but it's still very at the very, very beginning stage. When we start talking to some of these instrument vendors as well as hardware vendors, they actually are in the very exploring stage too. They have heard the buzzword, they have thought what this can do for them, but to marry their technology into that, honestly, I haven't really gotten anybody to give me concrete examples. Chad, I don't know how it is like for you guys, but for
Yeah.
me, that's mostly buzzwords.
Yeah, I would agree. And every new piece of software we look at now, somehow they tag AI on it. Everything runs by AI, right? Which is clearly not true. The one example in our company that's kind of interesting we've been looking at is how do we use AI in patient recruitment on the clinical side and identifying the right patients amongst the database. What are the types of people that will enroll in this trial and pre-selecting patients instead of trying to just bring them all in or call them and do the screens and all that. So if you can find the right patients that way by using an intelligent and learning system to do that and I don't want to and probably shouldn't divulge too many details, but we actually identified some guys right out of college that from a very well known university that were starting up a company, and I'm not even sure how we connected with them, but they connected with them through something maybe it was even LinkedIn, and they were like, yeah, we just brought these three young geniuses in that were like, yeah, we can chew this data up and they worked like they did 90% of the work in like one weekend. They're like, give us all the data, give it as clean as you can, but it doesn't need to be that clean because it's AI, and the AI is gonna chew that up and figure out how to process it, and then on the back end it spit out some data, and the initial outcome of that is really really positive as far as how it's looking.
Yeah, so they are I think a number of things that you can really work with. I would say implement. That's going to give you quite a bit of, I would say, saving.
You mentioned something, you said this could be revolutionary, but you're at the infancy. When you say revolutionary, what are you hoping or expecting it might do when you get there?
So I think I don't remember where I saw this, but there were this video I saw. I think was in a hospital. There was kind of like this robotic system that's kind of like a night prepping samples at night and doing some kind of analysis. I'm sure it's not mass spec, but it's what this robot that's kind of like working like has a wheel, then goes back and forth, take up a sample, did something to it, and did analysis. What we envision is eventually that, because ultimately right now the biological sample is still essentially prepped by human, one sample at a time, and they are um, go through various procedures we follow. Then we put that on the mass spec, one injection after another. My ultimate vision is have a learning robot that does the sample preparation on its own. I think that will require several generations of hardware and software before we can get there. One of the most important thing is that for example, what makes Tesla a very interesting computerized car is it's studded with camera and the LiDAR, like laser ranging. So really, that's the input of the system. You get the input, you get data, then the computer or AI does something with the data and self-learning, then there's an output. In Tesla's case, it can drive by itself because it knows how many cars in
Mm-hmm.
front, behind, around, what speed they're traveling at, what the road condition is. It can figure out if there's a deer across the road versus human, if it was really just the artifact of the light. If you look at the automation machineries for bioanalytical, none of them is there. Right now, the only thing maybe you can have in automation type of robots is sensing whether a sample is clogged, the tip is clogged, and it can go to a location. So there's a very few feedback sensors. Really what you need is to have the feedback sensors built into these automations. Then the data can become available for digestion by the computer or AI models. So it's going to take very coherent effort, both from the software engineer and the hardware engineer working together to, I imagine, to kind of reimagine what this automation machinery should look like. Just like I think McDonald's are going to start having robots that are going to flip burgers. I think eventually we are going to have robots that can essentially do sample prep.
It's just a matter of time.
Completely agree.
So, speaking of the regulatory side of the process, how do you intersect with and navigate the complexities of regulatory submissions in your role?
So one part of my job essentially is interact. First of all, there are a couple of parts. First part is we are in a regulated space, so we live under a set of guidances, both international and U.S. guidances. So we answer not only to FDA but also to other agencies across various geographical area, kind of like EMA, EMEA, the Chinese CFDA, the Japanese NMPA. So essentially, we work under the guidance of these organizations. So that's on one level, our work has to be compliant in a regulated space. We have to validate our assays to a standard that the agency says we need to be at. Actually, that's we also, as an industry, we get together, we talk about how to approach these issues. Chat runs a very large CRO, and actually they're in the exact same space. They follow the exact same type of guidances. So that's one part of my job is to kind of living under, make sure everybody, all the work we do, and all the people that works on my behalf, like CROs, follow those procedures that we have already established. The second part of it has to do with interacting with the agency when we do a submission. When we do a submission, we have to prove to the agency what we put into the documents have a scientific basis, has been validated, the numbers are accurate and precise. So in that, we produce a piece of documentation in the submission documents. That's our work. We basically form to the agency, these work are done under these standards, using these methods that have been scientifically proven to be sensitive, precise, and reproducible. Then the agency would review that and let us know if they feel there anything that is unbecoming, or they feel we need to improve, or they want a further explanation. So every single compound BMS file with the regulatory agency, that's going to have a biological component, goes through my department. So we really interact with agency in these two ways. The third way, more minor, is sometimes agency come knock on our door and audit us. So that's part of life a chat also has. When FDA or EMEA or whoever comes to the door, they want to make sure we're doing things exactly the way they expect. So that we have to handle the audits from agency, and that happens from time to time.